Hello, everybody. Good evening. So I'm Dr. Fernanda Pacheco. I'm an REI from Brazil and also the Director of Medical Affairs at Progenesis. And tonight I'm very excited to present this great webinar about deep dive into Progenesis Test-NIMA, which is a non-invasive metabolomic assay, and whole genome sequencing. We have two great speakers. First, Dr. Nabil Arrach, who is going to talk about NIMA test. Dr. Nabil Arrach has the interest in reducing the burden of genetic conditions during embryo development while improving the implantation and pregnancy rates for patients seeking fertility treatment, IVF, being the first scientist to develop next-generation sequencing for 24 chromosome aneuploidy screening, PGTA, and single genome disorder, PGTM. Dr. Arrach is constantly working on new ways to measure and predict embryo viability and implantation potential. He has PhD in molecular and cellular biology at the University of Seville, Spain, postdoctoral training in plant and microbiology department at UC Berkeley, postdoc in public health at the University of California at Berkeley, and MBA in San Diego State University. Dr. Nabil Arrach is the founder of Progenesis. He has published relevant papers that contribute to the research and development of PGT, and he has been the reviewer of Journal of Assisted Reproduction and Genetics since 2017. Afterwards, we are going to talk about whole genome sequencing and how it can overcome some challenges we have nowadays in reproductive genetics. And we are going to have this extraordinary speaker, Dr. Santiago Munne. Everybody knows Dr. Santiago Munne, but I'm going to review his CV. He got his PhD in genetics from Pittsburgh University. In 1993, he developed the first PGTA. He got many SART prizes because of this development, and also PGTA, PGTSR, and he demonstrated that PGTA reduced miscarriages and increased implantation rates. More SART prize in 1998. Santi has published more than 260 scientific publications, received SART and ASRN prize papers in 94, 95, 98, 2005, 2015, 16, 20, SRBT in 2022, and the ASRM Star Award consecutively from 2011 to 2022. In 2001, founded Reprogenetics with Jacques Cohen and David Sable, and as its CEO, Reprogenetics became the first commercial US PG lab performing over 100,000 procedures. Reprogenetics was sold in 2016 to Cooper Companies. In 2011, he co-founded with Alex Bizignano Recombine, which offered genetic carrier screening and was bought by Cooper Companies in 2016. These companies became Cooper Genomics, and Dr. Munne became its CSO. In 2017, He co-founded MedAnswers with CEO Alice Christie, an infertility telehealth platform. In 2017, he joined Martin Varsavsky and Jose Orcajodas to co-found Overture Life, a company focused on automating the IVF laboratory, which in 2023 published the first babies conceived with automated ICSI. Santi is a co-founder and president of Homo Health Ventures, an incubator for early-stage biomedical research specializing in reproductive medicine, which has invested in Sama Butterfly Bio, Gen Embryonics, among many other companies. He serves in several advisory boards of companies in reproductive medicine. As of January 2024, he is the scientific director of Progenesis. Welcome, Dr. Santiago Munne. So let's start with the talk about NIMA test with Dr. Nabil Arrach. And for the attendees, thank you for your presence. We have 45 participants, and we ask you to ask your questions on the Q&A tab, please. And at the end of the presentations, we are going to open the session for discussions.
Hi, today I'm very excited to be part of this webinar. It has been a long time. We have not personally presented a webinar on an exciting topic. Today we are having also Dr. Santiago privilege to be part of this webinar. My first talk is going to be about the NIMA test. NIMA stands for non-invasive metabolic assay. But before diving into the details, I would like to give a little bit of introduction of where we are. The status of PGTA stands as a gold standard in genetic testing. It has led to more than 95% accuracy in most of the cases. There's a lot of limitations due to quality of biopsy and other variables that are sometimes outside of the scope of the PGTA test. But one of the limitations I see that is most discussed among the IVF community is mosaicism. And mosaicism really presents a challenge for both patients and physicians. Mosaicism level varies dramatically from clinic to clinic, from embryologist to embryologist, from platform, sequencing platform to sequencing platform. And today there is no straightforward solution to that. Although some of the lab will report low mosaicism rates, our lab will report about 2%, but it's still a small amount of cases that you have to manage. The implantation rate with PGTA is about 17%. That means that 30% of the embryos that are transferred, we don't know why they're not transferred. We don't know if that is due to some limitation in the PGTA, or they are due to maternal factors, endometrial factors, or other factors that we are not aware. And so one of the most important drawbacks in PGTA is the time it takes for embryologists to biopsy the embryo and the risk associated with the biopsy. Biopsy is still a very invasive process, and the process of freezing and thawing add an additional level of stress to the embryo, which in this case sometimes not avoidable, not avoided. But at the end, the biopsy process is a time-consuming, expensive process, but also painful. Therefore, we have, and other groups explore non-invasive PGTA genomics assay, and it has been in the horizon for now up to 5 years. Lots of publications have emerged from different groups. However, the accuracy of non-invasive PGTA has always been a problem. Material contamination accounts for more than 10%, and gender discrepancy. I mean, embryos that are called females that are in fact male samples, and other limitations of false positive and false negatives. And so PGTA was the promise that we all always had as a substitute for biopsy. But the reality is it does not really reach in any way or any close to the accuracy of the biopsy. One of the ways to be able to improve the PGTA, non-invasive PGTA, is to make it more consistent among embryologists and among clinics across the media culture systems. That is very challenging problem to solve. In the recent years, we've tried the PGTA non-invasive test. Then we came up to the conclusion that somehow we should try a different way of looking at embryos. And so we have started to look at metabolism. The embryos that grow into culture media will consume the nutrients. They will release other metabolites, among which are peptide and cofactors and hormones. They will consume sugar and take it into a sugar metabolism and release byproducts. And in the media, you can also detect stress molecules and toxins and molecules that are problematic that can cause the embryo to fail the implantation. And so this approach avoids completely the material contamination. So there is no material contamination concepts in the metabolic test. Also, the consistency is very, very robust between samples. So the metabolism is a wonderful way of knowing if the embryo is growing according to the normal predictors. When we look at metabolites that are released in the media and we analyze them, we can come to certain conclusions that some metabolites correlates more with normal growth, while others correlate with abnormal, or indicate, it can be indicators of stress and developmental delay. So if you look at these nutshell, the comparison between non-invasive PGT and PGT and the NIMA, the non-invasive metabolic assay, you will see that most of the limitation we see with non-invasive PGT, which is, for example, requirements of consistency in the media, that requirement goes away with the metabolic tests because the samples requires very small amount of material and there is no dependency on gain. When you look at the sample quality, so the metabolic sample type really super exceed the expectation compared to the genomic assay tests. And the inconclusive results in the metabolic assay, you don't see there is always metabolites present in the media. So we're going to go into more details about the accuracy of the tests and how they compare to each other. For now, let's talk about the non-invasive metabolic assay. The assay uses a mass spec protocol to detect the type of molecules present in the media. This concept uses charge and mass. The samples are prepared in a matrix that provide ionization. The ionization will allow the sample, when it's bombarded by a laser, to release ions that are detected by a sensor. And when they reach the sensor, the time to fly determines the mass and the charge. So in general, a typical sample would have about 1,000 of molecules. We have focused on studying small molecules because it tends to be more informative which are both discrimination between euploid and aneuploid. And we have looked at the combining this technique with machine learning to try to process the data in a very efficient. So today, the fact that we're using genomics and using metabolomics and using AI offers a wonderful combination where you can use all these techniques into one approach. So you can use them in synergistic way to try to get the best information possible on embryos. So the AI approach is to look at images, whether they are static images or time-lapse images, and try to get the accuracy or prediction of what that embryo is, the prediction of diploidy, importation, and pregnancy. The problem with the relying on images only is that you can't really look at the entire story of the embryo. You're not looking at the DNA. You're not looking at the metabolism. You're just looking at growth and phases of growth, speed of growth, and you're looking at the structure of the embryo, which is good information, but is not enough to really tell the story of the embryo. By adding additional layers of genomics and metabolomics, you get the picture. One of the publications that was released on non-invasive assay talked about specific peaks that are indicative of a positive prognosis or negative peaks that are indicative of euploidy rotation pregnancy. The approach of looking at one specific peak or two few peaks is always a limiting factor because it forces you to look at certain markers that may not be present in embryos. We know that embryos have a fascinating way of dealing with stress and dealing with conditions, environmental conditions, and they can switch metabolic pathways. And so markers, a very narrow number of markers is not the right approach to analyze metabolomic data. It has to be an analysis, a comprehensive analysis, using machine learning, artificial intelligence to dissect the patterns that are indicative of a good result, good outcome. This is a typical pattern that you see, or typical profile you see with in spent media. In one column you see the media, in the other column you see the embryos. So if you try to identify peaks by eye, it will be virtually impossible. A typical sample will have about 1,000 peaks. Those peaks are ranging from a very small MZ, mass charge, to a mid-range of 1,000 MZ. This is a side-by-side comparing control and embryo. So in our study, we've looked at 200 patients, of which 150 were analyzed using mass spec screening method, and 50 were analyzed using a high mass spec analysis. So what we've seen is with those high resolution mass spec, you can identify the exact which is present and can be different comparing with unemployed or positive and negative. In this case, we focus just on euploid. The 150 samples went through mass spec. The data was extracted, normalized, and then it was run through machine using data training and testing. And this is the model that we have used. The model really relied on testing the samples, subset of samples, and then validating those samples in a new set of data. So using different models, we used models ranging from decision tree to random forest, which is fishing. And then we've seen that according to the model, we have had a range of high 80s, 86 to 92, 93% accuracy. But on average, the accuracy of the method was 90%. So we can tell at 80% if a sample is suitable and will annual based on the data set being tested. On the other data set that we have, high resolution data set we've identified 71 molecules that are unique to 15 molecules that are unique to any. And then we are now in a stage of looking at those specific molecules and understanding the biological function of each one and how we would provide. And we've also looked at molecules that can differentiate between sex, between male and female. We are currently working on enrolling new clinics in a new study to focus on molecular libraries that are potentially useful to differentiate between positive pregnancy to pregnancy and molecules that are indicative of biochemical loss. Artificial intelligence is a logical way to approach or to combine with existing diagnostics, with diagnostic methods. So the current status of AI, the use of AI in embryos, has been moderate. The success has been moderate, about 75%. And the reason is because, as I was mentioning earlier, the context of the data is not complete. It's missing diagnostics, missing genomic aspects, metabolic aspects, and also sometimes missing even the context of the patient, whether the BMI, the history of miscarriages, sometimes the stimulation protocol in response to certain drugs, etc. And so the ideal AI approach should take into consideration all these factors. And so if you look at current AI models that we use in embryo selection, they lead into about 80% accuracy. And so to really improve upon this is to be able to not only look at embryo images, but also add to that the diagnostics. So in this case, we have tested, we have developed an algorithms starting with embryo grading to predict the embryo grade of an embryo. And we have looked at the expansion ICM and TE. We have our accuracy ranging from 82% for expansion, 69% for ICM, 65% for tropical. So with this, means that we can combine all these parameters, all three parameters, and try to predict the grade of the embryo. But in reality, what we wanted to do is we wanted to be able to build an algorithm to predict the phases of the embryos and also to predict the UPOD imputation immunity from time lapse and from static images. But the idea is to try to combine all these methods into one approach in a way that you're looking at an embryo from genomics, from metabolomics, also from AI perspective. Taking all these three things into consideration, you're more likely to rank the embryo in a way that you can take advantage of the combinatory approach and choose the best embryo possible to lead to things. So this is just the early stage of the study. We are aiming to increase the number of participants, look at the bigger data sets, and look at pregnancy rates and implications. And hopefully in the next webinar, we will be able to provide you with additional data on these two. Finally, I'm going to talk about genes. Genes are genetics, embryology, and embryology symposium. It is organized in San Diego. This year is going to be May 29 to June 2nd. And it's a symposium that has really good quality speakers. Dr. Santiago Munne is our keynote speaker, and we are encouraging everyone to participate, attend, and be part of these exciting events and be part of the next generation of scientists. The last webinar, the last symposium we had last year was an excellent one, and we hope that this one will be even better. With this, I'm reaching the end of the presentation. Thank you so much.
Let me share my presentation.
Thank you. Thank you, Dr. Nabil. Yeah, please, Dr. Santiago.
Well, today I'm going to talk about whole genome sequencing of embryos. As you know, we can do PGT for several indications. So PGT for aneuploidy, in which usually we're using now low coverage sequencing. In this method, SNP analysis is needed to detect polyploidies, but otherwise we can detect all other chromosome abnormalities. We have PGT for structural rearrangements. Also, right now, most people are using low coverage sequencing to detect imbalances. However, if you want to detect balance translocations, then again, you'll have to use SNP analysis to differentiate carriers from normal embryos. Then we have PGTM. PGTM, for that, you need to detect SNP arrays or the mutation specifically. And one of the most used platforms is carrier mapping, but now there are a whole host of other platforms that are also trying to detect monogenetic diseases by analyzing either SNP arrays or the specific mutation, or through haplotyping of the embryos. And then we have also polygenic gene scores. Also, for that, you need a SNP analysis. And finally, which is one of the things that I'm going to talk today, is PGT, the de novo mutations. De novo mutations are mutations that are not present in the parents. Usually they are present, well, they are not present in the blood of the parents. They are present either in the germline of the parents, and these mutations accumulate with advancing maternal, but specifically or more commonly with paternal age. In order to detect them, since we have no idea if they are present or not in the parents, you really need to sequence the whole genome of the embryo. With that, you would have access to all the other PGT tests by analyzing every single letter of the genome of the embryo. So let's just start with some basic concepts. One is DNA amplification and their challenges. So there are several methods to amplify DNA. For instance, whole genome, shortplex, picoplex, and so on. These are used for copy number detection methods, but they produce very noisy profiles, and they are not very useful to detect SNP arrays. SNPs. Then you have to detect the SNPs. Mostly is use MDA or RepliG, but those also are not very good to detect copy number variables, and therefore also there are now newer methods such as Malbec, Lianti, and so on that can be used for detecting or amplifying DNA. On top of that, now more and more we're using complex bioinformatics to get rid of the noise that some of these methods are using. But in the past, mostly you were using PGTA, you would use whole genome amplification. Meanwhile, if you use, let's say for SNPs, for PGTM, you would use MDA or RepliG. Then the problem with DNA polymerase is that they are very prone to errors. These could be errors of for instance, deamination. And the problem is that once the error occurs, by the amplification, this error is amplified. And so you have more copies of that error going down. A way of avoiding or detecting that this is the case is by analyzing both strands of the DNA and then just using the ones that are complementary. So if you have the same result for both strands of DNA, then you know that there is no errors there. So right now there are a bunch of methods that are trying to combine the detection of PGT-A, M, and structural abnormalities in a single platform or in a single method, although some use a combination of platforms. For instance, there are a bunch of methods that are using SNP arrays to detect everything, others that only use sequencing, and others that use a combination. So usually they use sequencing and then they amplify some SNPs specific for the disease. I will cover only a couple of those that are mostly used, which are the ones in green. So for instance, sequencing strategies for comprehensive PGT. One of the first ones was PGT Complete by Cooper. Basically, they use NovaSeq, which they can read 16 million reads of sequencing, and therefore by that you get a lot of SNPs also. So you are not only doing quantitative analysis, but also you get a lot of information on SNPs, and with that you can combine copy numbers, which will be quantitative, and SNPs, which will be qualitative. Also, they use per-10 sequencing, and which, as I said, avoids some of the errors that happen during amplification. And they use artificial intelligence to do the analysis automatically. With that, they were able to reduce the amount of mosaics, and the analysis and reporting is automatic, and they can detect all triploidies, and also give parental quality control to see if the embryo is from the parents or not. It has not been validated, as far as I know, for UPD, balanced translocations, or for PGT-M, but in theory, since they are detecting SNPs, you could do haplotyping with that, and you'll be able to use it for that purpose. As I said, I haven't seen yet the data for that. With that, they published that they reduced the amount of mosaics that they obtain, and that it was significantly better than the platform that they had before. Another method by Masset et al. Is called single-cell genotyping by sequencing. Basically, also they use here, they are also analyzing the DNA of the parents. So then with also quite in-depth sequencing, 7x depth, then you can do haplotyping of the parents, meaning that you would know from which parent that abnormality comes from, which SNPs come from which parent, and that will allow you then to do PGT-A just by counting, and also PGT-M because you have the haplotype of the parents, and you would know where the mutation in the chromosome of the specific parent is. Then you have haplotyping by whole genome sequencing, a very similar method also. Here they use 4x depth for embryos and 10 for parents. And in theory also it could detect PGT-A, structural abnormalities, and PGT-M. Another one is S-haploseq. This is very new. Again, haplotyping by whole genome sequencing. The difference with this one and the previous is that it can sequence the embryos at very low resolution, so only 0.4 x depth, and the parents at 4x. And then has some algorithms that will infer the haplotypes of the parents, and with that you can do PGT-M. Also, the advantage is that if you don't need PGT-M, you don't need to do the sequencing of the parents, and then it's much cheaper. Then there are strategies that do genotyping, so they analyze the SNPs directly to do comprehensive PGT. One of the first ones was high-resolution SNP arrays by the team of Nathan Treff. Basically, they use high-resolution SNP arrays that they have 800,000 SNPs. And it can detect PGTA, M, SR, and also PGTP. They claim that the resolution it's up to 1 megabase in size, so they can detect abnormalities up to 1 megabase. Sorry, 1 megabase and above. They just published a paper recently detecting CNVs. So, for instance, 1.6% of babies or pregnancies have microdeletions or microduplications syndromes. And about 34% of them are inherited. So what they did is go back to all the cases that they have analyzed with this platform, and they detected that in 0.7% of them, there were more than one embryo that had exactly the same CNV. And then they analyzed the parents, and in fact, they detected that those parents had either translocations or some pathogenic copy number variables. So with this platform, they are basically detecting 0.7 of the embryos have CNVs that were not identified in the parents because the parents were not analyzed at that level. Also, the platform can detect polygenic risk scores. Polygenic risk scores are basically are used for diseases that are multigenic. So there are, for instance, breast cancer, type 2 diabetes, etc. They have dozens to thousands of genes implicated in that disease. And therefore, you need to analyze all those and then come up with a risk score, because obviously the combinations are like a Gauss bell. So depending where you are in that bell, you have more or less risk of that disease. So, for instance, they think that they can reduce significantly by choosing by using this method, some of the risk from 45 to 71% for some specific diseases of these complex diseases. Another method that also it's SNP-based is haplorhythmicis. Here again, they amplify the parents and the embryos, and they use SNP genotyping, and then they calculate log R and B allele frequencies to do the haplotyping of the embryos. And with that, you can detect a copy number, and also you can detect mutations by inferring them from the chromosomes, where what's the origin of that chromosome. But to the subject of my talk, which is PGT de novo mutations. So the novo mutations, they accumulate in the parents' gametes or occur at embryogenesis, but they cannot be detected in the parents. We think that there are 74 de novo SNP mutations per embryo. Not all, obviously, not all of them are pathogenic, and some will be in introns that in exons that in introns that will not code for anything, but they are frequent. And these de novo mutations cannot be detected, obviously, by carrier screening because they are not detected in the blood. They are in the parents' gametes. You won't find them in the saliva or in blood. So we don't know a priori that this couple has a de novo mutation, or in their sperm or eggs they have de novo mutations. De novo mutations increase with paternal age significantly. For instance, a father that is 45 years old have 3.5 more risk of autism than a young one, or 27 times more risk of bipolar disorders than somebody that's, let's say, 24 years old. So they increase dramatically with paternal age. And obviously, if we're the average female that we're treating in IVF now, it's about 35. Probably the guys are 35 and above, 40. So the risk of these mutations is not small. The Genome mutations are more detrimental also than inherited mutations, since they have not been exposed to natural selection. So the ones that are inherited, usually they have some positive effect in heterogeneous complement. So for instance, well, there are a bunch of diseases that in heterogeneous form gives you some resistance. For instance, sickle cell anemia gives you resistant to malaria, but obviously if you have the two deleterious mutations, then you'll be affected. But the no mutations, since they have not been through natural selection, they are new, they are much more extreme in the damage that they cause. The number of mutations are the primary cause of congenital malformation syndromes, severe intellectual disability, autism, spectral disorders, schizophrenia, and they cause 48 to 63% of fetal malformations. Finally, people are starting to realize that also men have a biological clock. As I said, these mutations increase with paternal age, also with maternal age, but specifically or more often with paternal age. Also, it's not only point mutations. We can have copy number variants, so they are larger. And it is known that these are extremely unlikely that they are produced by amplification artifacts, obviously, unless they are in the form of mosaicism. And they affect 1 to 2% of conceptions. And what is seen is that the more CMVs per embryo or per baby, the higher the chance that they would produce autism. Another problem could be that, for instance, if we don't do PGT, you know that there is a significant decrease in ongoing pregnancy rate without PGT. Then if we do PGT, there is a slight decrease in implantation potential with maternal age. It's a small, almost, if you transfer a euploid embryo, almost they implant equally well at any age. There is a small decrease after 41, 42, which is for unknown reasons. We don't know why. But then you have still 40% of embryos on average that even if they are euploid, they don't implant. And some of those could be caused by de novo mutations. So there are at least 3 methods described. Well, no, sorry, there are only 3 methods described to do whole genome sequencing of embryos. There are now a couple of companies claiming that they can do it, but they haven't shown any papers, so we don't know if this is for real. So the first one was Peters et al., and I was part of also of that study. And basically, they individually sequence 384 DNA fragments to allow haplotyping and detect amplification artifacts. The other one is by Xie et al., published recently, that uses PTA amplification, which is a new way of amplifying DNA that is more faithful. And the other is by Murphy et al., which uses standard amplification but has very complex bioinformatics filtering to detect false positives. So the first one was called large fragment reads, or use large fragment reads. Basically, the DNA was dispersed into 384 compartments, and therefore each one of them had only about 5% haploid genome in each. And then each one of them, of these compartments, is amplified, fragmented, and barcoded independently of the others. And therefore, then you can compare them And if you have the same results in several of the compartments for the same region, then you would know that there is no artifact and that this is real, right? So with that technique, we were able to detect 97% of the embryo genome, and we found 3,000 de novo mutations or variants per embryo. Obviously, most of them were variants of unknown significance or not pathogenic, but 30 were pathogenic. Then the other method is primary template direct amplification, or PTA, and it uses MDA polymerase plus isomerase resistant terminators that create smaller amplicons. So the pieces of DNA that are amplified are smaller. And the amplicons undergo limited subsequent amplification with more of the amplicon occurring from the primary template. So let's say if there is a mistake in the amplification, since here you are amplifying more from the initial template, the errors propagate less. So any error in the daughter amplicon have limited propagation during the following amplification. If you can see at the bottom, you could see how noisy is the MDA profile compared to the PTA profile. And with this method, it seems to amplify much better than any other method. And it has 90% detection coverage in single cells, so much better than other methods. They have done very limited analysis of embryos. So basically, they did only analyze 4 embryos, and they compared 2 biopsies, and they obtained 96% genome coverage at 14 depths, and it detected 3 million SNVs, and 96% of the 2 biopsies were concordant. Still, that means that there were 70,000 SNPs that were different. So they still have quite a bit of work polishing these results. In theory, it can detect aneuploidy, inherited diseases, and de novo mutations, and polygenic risk scores. The third method is by Murphy et al. And this uses a regular whole genome amplification, but then it sequences a much higher depth, so 30x for parents and 50 depth for the embryo. And then it uses a lot of variant filtration to detect false positives. And once the filtration has occurred, then it annotates the variants based on 50 different sources of information. It can detect de novo mutations, inherited mutations, aneuploidy, translocations, triple nucleotide repeats, and polygenic gene scores. So it's just an example of how they filter. This is the pipeline of filtration of artifacts. And this is another pipeline to detect all these artifacts. As you can see, it's all bioinformatics to clean the data from errors. And once it's filtered, then they try to analyze if this variant is pathogenic or not, comparing to these 50 or more databases that have been published so far. So basically, they detect about 200 to 500,000 false positives. So this is 5 to 15%, which is obviously too high. And these errors are introduced by base misincorporation errors, sequencing errors, alignment errors. But then with the filtering, they can reduce this to 90% or more. So for instance, in order to do the validation, they use genome in a bottle, NA12878, which is a very well-studied control, and then compare genomic DNA to amplified DNA. and then assemblies and ploidy were assessed via transmission and read binding for copy number, and tandem repeat disorders reported via carrier repeat numbers. The number of mutations accuracy is calculated after the filtering, and annotation improves accuracy significantly. The number of mutations after annotation are reconfirmed by another platform, by Sanger. The positive predictive value was for SNPs was 98%, and for indels 93%. The negative predictive value was very high, almost 100% for both. Sensitivity 92% for SNPs, specificity almost 100% for SNPs, and accuracy also 99%. The false positive rate was 0.2% for SNPs. So basically, these are the pre-annotation de novo mutations that were detected, either in the amplified DNA or in the biopsy model. The biopsy model were basically 5 cells put in tubes of these control DNA. And post-annotation, they were reduced to only 1.17 pathogenics per sample. So a significant reduction. So from 3,000 pre-annotation, after the filtering, we reach only 1.2 de novo mutations per embryo. So this shows you that this method is quite it cleans all the noise that all the amplification created. The previous ones, or the second one, had like 70,000 de novo mutations. This here, we reduce it to 1.2 per embryo. It's still a lot, right? Every embryo could have a de novo mutation, and obviously some of those will be pathogenic. So obviously the question is if this will be very expensive. We're sequencing at 50x. But as you know, the price of sequencing it's decreasing dramatically. Right now, with this new platform, it can cost you only 200 per embryo. And then the strategy obviously is to do first PGTA. That will eliminate 50% of the embryos, and only do whole genome sequencing for the embryos that are euploid, with which then the cost is not so high. Obviously, the other question is how you counsel these patients. And we have 2 approaches. One is, and I think this is the more conductive for IVF centers, it's only to report pathogenic diseases or pathogenic variables. And this is based also on what is done with prenatal diagnosis. So whatever you don't know what's the effect, you don't report it, only report whatever is pathogenic. And then you can use, let's say, a reference lab, and we send you a report, a normal report, in which it says this embryo is aneuploid, this other embryo has a pathogenic mutation, so don't transfer either of those. The other approach, obviously, is that you have information on the whole genome, and you could spend hours and hours with the couple, telling them if this embryo is going to go to Harvard, or it will be better to go to the NFL. And this is a completely different approach of counseling. And some companies are edging in that direction. Usually these are companies that have been generated in Silicon Valley or so. But for those companies or those PGT labs that are in our field, I think the first approach is right now the more sensitive one. And with that, I will announce here for the first time that Progenesis and Gene Embryonics are going to collaborate, and Progenesis is going to offer this service, PGT, with whole genome amplification in the US, LATAM, and India. So in conclusion, it took us 30 years to go from 5 chromosomes to whole genome sequencing of embryos. And almost 70% of euploid embryos implant, but still there are 30% that we don't know why they don't implant. It could be metabolomics, it could be mutations, and so on. So combining all these methods, hopefully we'll be able to identify why or we'll be able to reach 100% implantation. There is a variety of platforms that can do simultaneously PGT, MSR, PS, but only whole genome sequencing can detect de novo mutations, which actually happen to be the most common mutations found in babies. And the PGT lab right now is fully automated, but still we have a limitation, which is that the IVF lab is not yet fully automated. So also there are some, I think the biopsy process is the weakened link here. So either non-invasive PGT or automated biopsies may standardize this process. And well, thank you very much for your attention. And if you have any specific questions that you don't want to ask now and you want to ask me directly, here is my email. Thank you very much.
Thank you very much, Dr. Santiago, for your excellent presentation. So now I'm going to open for questions from our attendees. And I want to announce that at the very end of this webinar, we are going to have a raffle, and the winner is going to win a free registration for GENES, our symposium that's going to happen in San Diego at the end of May, from May 30th to June 1st in San Diego. So let me see. Okay, I invite you all to ask questions. Dr. Santiago, while I wait for questions from our attendees, I want to again congratulate for the excellent presentation, very exciting. And I want to know, how do you feel to see all the development of PGT? Since you were at the very beginning.
Yeah. As I said, it took 30 years to arrive here.
Yeah.
And still there is quite a bit of debate if PGT works or not. Obviously it does. But I think what I said, the limitation that we have is that although the PGT lab could be fully automated, there are still a lot of variability in the IVF lab. And if you do things not correctly, then it's easy to say that things don't work. Meanwhile, if we have standard platform, let's say automated, some of the automated processes in the IVF lab, then we will have less errors, and it will be easy to progress and innovate further if we have platforms from which everybody gets the same results, right?
Yes. And how do you see the variation that occurs also in the PGT labs? And
yes,
with all these platforms and protocols and different algorithms.
Yes, obviously this depends on, I mean, almost everybody is using the same platforms. Well, either SNP arrays or sequencing, and it depends obviously on the depth of sequencing that we're using and the level of automation. But most of the platforms right now are I would say, getting the same results. The only problem is mosaicism, how you report it. Obviously, 3-4 years ago, 5 years ago, we didn't know what was the implication of mosaicism. Now there is plenty of data that show that low-level mosaics is they implant equally well as euploid embryos. So we shouldn't even have to report it because some of that is noise. And once this is agreed, I think there are not too many differences between platforms. Obviously, the more sequencing depth you add, the more things you can start detecting. But also the cost of the test increases. Hopefully, as I said, with decreasing sequencing cost, this would not be a problem.
Okay, thank you very much. And me as an REI, I'm very excited to see the development of IVF and how we can evaluate the best embryo, the most viable embryo for implantation beyond genomics, how we are learning so much with all the omics, metabolomics, proteomics. And I believe NIMA test and AI and also, you know, adding all these tests are going to increase accuracy. Every time we add more tests, we increase accuracy. So I'm very excited to see the next 10 years in IVF. I think we'll change a lot with all this technology, and also how we are understanding more and more about the embryo genomics and how it can help families to have healthy babies. So this is very exciting. I want to know if Dr. Nabil is in this call, if he wants to say something. I believe he is in the call. I heard he was in the call. I'm so sorry, Dr. Nabil had
Yeah, I am. I am on the call as well. I apologize as I am in an airport, so it may be a little bit noisy. No, I
Okay.
will just emphasize what you have said, that we are in the era of the fusion between artificial intelligence and diagnostics, and those two fields are now in the verse of being merged into one approach, one multifactorial approach that looks at the embryo from multiple angles. We're not relying on DNA only to tell us whether the embryo is fit for transfer and pregnancy. We're looking at other contexts. We're looking at the kinetic of the embryo. We're looking at the images, time lapse, and then we're looking at metabolic profiling. And with this exciting development in whole genome sequencing, we're going to look at things that we have never looked at, things that may explain why embryos fail in those 30% of implantation. And there are lots of translocation and inversions that happen during meiosis that we never know about them. They may happen even in, they may cause miscarriages, and they may cause implantation failure that we would never be able to catch. And so I feel like I share the same sentiments. I feel like we're in a in a tip point situation where we're going to definitely see a jump in sensitivity and technology.
Awesome. Dr. Nabil, I will take one more question for you. It comes from Chihan. Chihan is the lab director at Yale, and he asks, how are the samples for metabolomic analysis prepared to send out? Can the same sample be used for non-invasive PGT?
The samples are used in the same way they would be used for non-invasive PGT. There are samples collected from media, for culture media. We encourage or recommend that there is a change of the media in day 3 or day 4, and the collection could happen day 5 or day 6. The beauty about these tests is it can adjust to the culture system of a clinic if needed. Meaning if the clinic has a particular way of growing the embryos, as long as they have large number of samples, the algorithms can adjust because it's a diagnostic test, but also fused with machine learning. So it does collect data on media and data on embryos and data on clinical outcome and may adjust over time. But the straightforward answer would be no different. The media preparation is no different from the non-invasive PGT, and in fact, the same media can be used for both if needed.
Okay, Dr. Nabil, and can you tell the audience how is the NIMA test being what's the dateline for the NIMA test? We have already the validation. We are waiting for testing the algorithm. How is the NIMA test in the?
So currently we have validated the test using 200 patients. It's a small data set, but the validation was based on the concordance between metabolic profile and a biopsy results, the PGT biopsy results. In this next phase, we are enrolling more clinics to grow the number of samples and the diversity in culture media, culture systems, and culture media. And we're going to look at the correlation between the metabolic profile and implantation and pregnancy rates. I have to add that we have also identified certain molecules that may give us additional information on sex of the embryo, but this is still an early stage. But we have some candidate molecules that can also be informative if sex is important in that selection. And so just to summarize, we're going to go for a second phase of a multi-center study. And while preparing for, after that, we will prepare for launch.
Awesome. Thank you very much. And again, with Dr. Santi now, it was so interesting for your presentation that you mentioned that, of course, de novo mutations is more related to paternal age, but also maternal age. But studies have shown more evidence related to paternal age. And we see that more and more men with above 40 or 50, they become a father again. Or for the first time. So, and those de novo mutations can be related to clinical pathology as autism and other neurological development disorders. So this is very important, as you mentioned, because it's becoming more and more common. And how interesting it is, this data that you brought, saying that about 45–50% of all malformation in babies come from de novo mutations that didn't exist. We couldn't identify in the parents. So that's really impressive. Thank you for bringing us this data.
Yes, this is, I think, the last thing that PCT needs to solve. And now I think it's solved. Obviously, we need to see more data, and we're going to get that data. But yeah, that's the last frontier, basically analyzing the whole genome of the embryo.
Yes, because many patients, they still have the thought that a PGT-A, a simple PGT-A, is going to show all pathologies, all genomic pathologies in their babies. And unfortunately, it's not like this, as all tests, there are their limitations, but it's good that this field is developing, and so we still have a lot to learn, right? So, and this is awesome to help families to build healthy families, right? So, very exciting. So, I don't have more questions, unfortunately. I don't know, Dr. Santi, if you want to speak something to finish your collaboration in this webinar.
Yeah, no, just welcome also everybody to the meeting in San Diego, Genes. It's going to be very interesting as the last one. So we hope that you will join us in that meeting.
Awesome, awesome. Thank you very much. And if you want, you stay with us for the raffle. If not, you can say goodbye. And we are going to do this raffle. Bye-bye. Thank you very much for your presence.
Thank you so much.
Thank you. Thank you, Dr. Nabil. We are going to share a QR code where all attendees can register for the symposium. Unfortunately, though, this symposium is just for IVF professionals, physicians, embryologists, genetic counselors. So unfortunately, patients can't participate, but on your right, you see the QR code for registration if you want to register. And for the raffle,
yeah, we'll go ahead and do a raffle. So we individually selected, and that person came out to be Jack Crane. So you'll be receiving an email from Genes with your free registration. So look for that after this webinar concludes.
oh, okay. So they did the raffle already. I'm so sorry. I thought they were going to show a QR code, and then you would register on a Google survey and do the raffle tomorrow. But okay, so congratulations. We will reach out to you to give more information. Thank you, Thomas, for the clarification. And here more about Genes. We have our website, app, the QR code for registration, also this small QR code, the black one, is to subscribe for our email and receive more information about Genes. We also have some events as cocktail and dinner, lunch, also dinner at a yacht. It was wonderful last year. So we are going to repeat this yacht event, and you are all invited for this amazing symposium. I have received some questions here. If NIMA will reveal 7-day embryo, or is that clinic specific? You are still able to register using the QR code. Okay, Lindsay answer. So we are going to answer to you, okay, Sabrina Kinsella. And so I'm going to finish this webinar. I thank you, Lindsay and Thomas. They work in our marketing department, and they organized all the webinar. So I thank you very much to host this webinar with me. And I thank you all our attendees to participate. And we are going to have the next webinar on March 13th. We usually have those webinars on the second Wednesday of each month. This month was an exception because our speakers calendar, but every second Wednesday of each month we have Progenesis Academy, and our goal is to promote debate, discussion, and awareness about multiple topics in IVF, reproductive genetics, for the community and patients. Okay, thank you very much for your presence.